My name is Max Grassmann and I'm professor of physiology, of human and veterinary physiology at the University of Zurich. I'm interested in how the body reacts to reduced oxygenation. This means how our cells cope with hypoxic conditions. So I'm interested in respiration and in the lung. And of note, when it comes to the lung, there is one name you cannot miss. This is Ewald Weibel. I met Ewald back in 1992, and ever since he has been a bright, successful, and very helpful mentor during all these years. But who is Ewald Weibel? Who is this man who established lung morphology, who at the age of 88 still publishes papers and has been very successful so far with citations of 25,000 and more? Let me guide you through the most remarkable life of Ewald Weibel, this incredible mentor. Ewald, where are we?
Well, thank you very much, Max. I hope I can cope with your expectations. Well, this is the old auditorium of the Institute of Anatomy at the University of Bern in Switzerland. It was built in 1896 as an anatomical theater in the style of medieval medicine, patterned so that it is ideal for the students to watch either things on the blackboard or on the dissection table, the main teaching methods at the time. It is not very comfortable to sit in, as you notice, so I had to fight for over 20 years the architects who wanted to renovate it, because I felt this is a perfect auditorium. It offers very good sight. As I said before, it has good acoustics, and perhaps this is the most important, it offers good personal face-to-face contact between the students and the teacher.
But wait a minute, Ewald. Anatomy? So how comes that I interview you here for the American Physiological Society?
Well, it's easy. I earned my bread all life by teaching anatomy to medical students. But in my way of thinking, I think more like a physiologist. But I understand that this is a big problem, because when I was elected president of the International Union of Physiological Sciences in 1996, our chairman of physiology here in Bern quipped, "How deep has physiology fallen that we now have to have an anatomist as our president?" Well, I have no problems with that, because for me, morphology or anatomy is an integral part, an important part of the physiological sciences in the broader sense.
So the APS meetings were most important for you compared to the anatomical ones?
Well, yes. I mean, I attended almost all APS meetings since 1961, when I gave my first presentation on lung morphometry under the chairmanship of Robert Forster in Atlantic City at the FASEP meeting. But I must say that I have never attended a meeting of the American Association of Anatomists. And when I was made an honorary member of the APS, I was very proud. Of course, I'm not an honorary member of the American Anatomists.
So, Ewald, let's go back to your roots. You are Swiss, and you grew up in Switzerland during the Second World War?
I grew up in Aarau, which is in the middle, in the heart of Switzerland, halfway between Zurich and Bern. When I was 10 years old, the Second World War broke out, and, well, of course, Switzerland was not involved in the war because we were neutral, but, of course, there was fear of what was happening all around us. There were some shortages, but life went on. I grew up in a non-academic middle-class family. My father was a typewriter mechanic. It was a very skilled job in the times of the mechanical typewriters. And my mother was a seamstress. But that was really no obstacle to a good education. We had a wonderful school system in Switzerland which fostered the students according to their potential, I easily made it to the gymnasium, and then it was our family doctor who suggested I might study medicine. First of all, because it would cover, well, the broad interests I had, which went from science, nature to culture and art, but mainly it would ensure a job and a salary. So I went to the medical school in Zurich, and in fact the state paid a stipend to cover my tuition at the University of Zurich.
So when you were there, as I remember that correctly from all your publications and discussions we had, did your love for science already started at the University of Zurich when you were a medical student?
No, actually earlier. I attended in Aarau the Cantonal College, the same school that Albert Einstein had attended 50 years before me. It is and was and is a wonderful school where the teachers challenging the students. I was particularly influenced by two of the professors, one the biologist and the other one the mathematician. The biologist for his great didactic skills and the mathematician for his rigorous thinking. The school challenged us to the point that we were actually stimulated to ask for more questions that really were in the curriculum, and so the teachers partly gave us special courses, so for example on calculus, because our class, that was a very energetic class, asked for that. Well, as an aside, our little class of 24 generated 2 members of the US National Academy of Sciences, besides myself, Werner Arber, who had won the Nobel Prize in 1978. In medical school in Zurich, I was particularly fascinated by anatomy and physiology. Anatomy for the beauty of structure, physiology for the rigorous regulatory processes that were analyzed. And because of my big interaction with teachers at the college time, I also had the courage of challenging our professor of physiology in Zurich on the theory of hearing that he was telling us. He was Walter Hess, who had won the Nobel Prize for his work on autonomic regulation. So I approached him and said I did not believe in the theory of hearing, whether he would support a thesis work on the physiology of hearing. And he said, no, there is no sense doing any physiology on the hearing process before we know more about the structure of the inner ear. Go to the anatomist. So I did, and I did my thesis work on the development of the cochlear duct, measuring basilar membranes and its different parts. So that was my step from physiology to anatomy.
But from the ear, that's a very long way to come to the lung.
Well, that was opportunism. After graduation from medical school, I had to have a paid job. My parents could not further support me. So I first worked as assistant in anatomy, finishing my thesis. And my plan was to go then through pathology into surgery. And we must know that in those days, one did not get a salary for the first 2 years of clinical training. And I could not afford that. So the plan, go through pathology, might perhaps solve this. But the professor of pathology himself had a condition to give me a modest little salary. And this is that I should study the anastomosis between the bronchial and the pulmonary artery in the human lung while I was in anatomy. And particularly study the regulatory vessels of these anastomoses, which was a hype at the time, the so-called Sperrarterien that would be able to close up the blood flow.
So you did that work and finished that in Zurich.
Yes, I looked at human lungs, studied the distribution of anastomosis in serial sections, and found that they occurred everywhere in the lung, from the top to the bottom. But when I looked for these Sperrarterien, these were arteries with a longitudinal muscle bundle in the intima, they occurred only in the lower lobe. So clearly, it appeared to me, and I was a young Turk then, that this had nothing to do with the regulation of blood flow. So perhaps this was simply a protective reaction to stretching of arteries by the ventilation process, which would be greater in the lower lobe. So I did a little experiment, took some rats, stitched an intestinal loop up to the diaphragm in such a way that one of the mesenteric arteries would be subjected to the traction of the ventilatory movement. And sure enough, after 3 weeks, these arteries had developed a wonderful bundle of longitudinal fibers in their intima. And that, of course, was the end of Sperrarterien as regulatory vessels.
So you killed the Sperrarterien theory at that time and then went as a postdoctoral fellow to the US?
Well, because of this success, I was invited to apply for a fellowship, 2-year fellowship, co-sponsored by the Swiss Medical Academy and NIH. And so I had to choose a topic, and I wanted to study the physiology of anastomosis between the bronchial and the pulmonary arteries, what was called collateral circulation. So my first choice was André Cournand at Columbia University, who was doing some studies in that field, and who had just, well, this was 1957, had just in 1956 received the Nobel Prize for his work on cardiac catheterization. So I wrote him a letter with documents, never got a reply. So I approached my second choice, Avery Lebo, the pathologist at Yale University, who immediately accepted me so I could file my proposal before the deadline. But many weeks later, my letter to Cournand bounced back, addressee unknown.
So they didn't know their own Nobel Prize winner?
They did not even, were not even able to locate their latest Nobel Prize winner. So in June 1958, the fellowship was approved. I traveled by a freighter ship with my young wife to New York, and then up to New Haven to take my new job in the lab of Lebo.
And how was the work in this brand new lab?
Fascinating. Very different from what I had been used to before. There was lively work, there was teamwork around the boss. We all assisted him when he did his experiments, but each had his own project. I had a project on studying the development of collateral circulation after ligation of a pulmonary artery. And then, about a few months after my arrival in New Haven, I received a letter from Cournand, who invited me to come and give a seminar on the regulation of the anastomosis that is on the Sperrarterien. This should be in February 1959 at Columbia University in his laboratory. And at the end of that, that was, by the way, my first seminar I gave in English. So at the end of the seminar, Cournand, together with Dickinson Richards, who was the head of the pulmonary division at Bellevue Hospital, told me, "We want you to come and work with us." So when I shyly asked, what would you like a young Swiss anatomist to do in your physiology lab? They simply answered, "Do anything on the structure of the lung that is of interest for physiology." Well, that was almost too good to be true. And I realized this was a lifetime project, and I accepted on the spot.
So you switched the labs immediately and went to your first choice lab?
Well, not really. I mean, I had an obligation to my fellowship, but I finished my project in 1 year instead of 2. In June '59, I handed a manuscript, also my first manuscript in English, to Ewald Weibel, and he had it published in Circulation Research the same year.
And how did you move them from one lab to the other?
Well, then I went first for a trip to through the United States with my wife on our old Nash Statesman 1951 that we had bought for $200. And so we went to Grand Canyon, California, and all the way back to New York to take on the new job.
And then you started?
Then I started on the 1st of September 1959. I had 2 labs, generous funds to equipment, but I had no project.
But you could do anything you like to do that brings you on the structure of the lung, that brings physiology further.
Yes, but what was it? My worry was, what have we anatomists missed in our work, that our work is not of interest for physiology? But the answer came during the first weeks I was at Bellevue Hospital, and it came from another newcomer to the lab, Domingo Gomez, a Cuban cardiologist who had been jailed by Fidel Castro in his revolution earlier that year, could escape and flee to New York, and was given refuge by Cournand.
So the Cuban researcher really influenced on your way of thinking for the next years?
Yes, very much so. In fact, well, maybe I should say who Gomez was. Gomez was born in Cuba in 1903, so he was the age of my father, as a son of a peasant. He got his first education for his grandmother, and then with the help of an American country doctor who worked in Cuba, he somehow got into the medical school in Havana. And because he was a particularly bright guy, he was selected to go to Paris to finish his medical school at the École de Médecine in 1928. And parallel to that, he also studied mathematics at the Sorbonne and graduated in it also. Then he worked as a cardiologist. He stayed in Paris and worked as a cardiologist in various university hospitals. But he was mainly a brilliant biophysicist. In 1941, he published a book, 730 pages, 613 equations on angiogenetics and hemodynamics in the human circulation. But a year after that, he had to flee France because of the Nazi invasion of Paris, and he went to the United States, to New York, where he got a job at New York University in the Department of Physiology with Homer Smith. But then in 1953, Batista, the dictator of Cuba, called him back to Cuba with the task to establish a cardiac institute in Cuba. But that actually never materialized because of the political turmoils which then ended in Fidel Castro's revolution in 1959 and forced Gomez to flee again, this time from Cuba to America, and then he was given refuge by Cournand at Columbia University. Cournand had known him from the time at NYU.
So with this biophysical background of Domingo Gomez, what was his project in the lab? Well,
Cournand had given him an assistant professorship, which gave him a living, and a tiny little office. And Gomez had then decided he would now concentrate on the lung because of the environment he was in, studying aerodynamics and gas exchange, trying to model it by first principles in the sense of a theoretical physicist. And for that, he needed information on lung structure. And so I was a welcome partner. We had many coffee breaks, and in one of them he simply asked me, how many alveoli are there in the lung? I said, no. So I looked up in the literature and found there were 66 million, in another paper 1.6 billion alveoli. Of course, all guesstimates.
So what were the problems?
Well, the problem was that these tiny little structures, alveoli, a quarter millimeter in diameter, could only be seen in the microscope on thin sections. And there was no way of counting such little structures from counts on the section to be relevant for the three-dimensional space. So Gomez and I simply invented a method. We invented a method on the basis of geometric probability considerations of cutting an alveolus in various directions by a random section. He believed in the method, and I was skeptical, so I wanted to test it by a little experiment. I took some peas, embedded them in gelatin, let it solidify, sliced it, and then counted the profiles on the slices. And, well, got the same number as what I had put in, so the method worked. And we applied it to the human lung, found that an adult human lung contains about 300 million alveoli. But then while I was doing this work with the p-aspic, messy work, Cournand enters my lab. And he stares at me and says, "What are you doing? I did not hire you to study p-aspic," left the room in anger. Well, the point was that Cournand was, because of his Nobel Prize, a very much demanded speaker all over the country. And when he came back from one of his teaching trips, he would go through the labs, assemble his collaborators in the conference room, and look for new information, new slides for his next lectures. And my p-aspic evidently was not an attractive object. But I must say that Cournand was appeased later when the picture of the p-aspic was published in the Journal of Applied Physiology, one of the most renowned physiology journals, and probably this was the only picture of vegetable ever published in this journal.
So nevertheless, peas basically solved you the problem.
Well, not really, because Gomez was not interested in the number of alveoli. This was a number for anatomists. Gomez wanted to have the number of alveoli to calculate the alveolar surface area as the main determinant of gas exchange. But it turned out that, in fact, one could also obtain these measurements in a different way, by a direct statistical sampling procedure with lines or needles, if you want, counting the number of intersections, something that came up in the course of time, rather early in the game, because in fact I came upon this problem of obtaining measurements on sections that would be relevant for the three-dimensional structure at a very good time. Around 1960, this problem arose also in other fields, for example in material sciences, alloys, or in geology, and also in biology by other people. So particularly Hans Elias, the anatomist from Chicago, was keenly interested in that. I knew him from before. And he actually coined the term stereology for this kind of approach of studying three-dimensional structures on sections. And he founded an international society for stereology, a small society, but a very effective one, which brought forward this new approach of doing measurements on sections that were relevant in a rather rapid time. And I was fortunate to be part of that group.
So stereology solves all the problems to do the structural work so that you could finally work on the physiology of the lung?
Well, not really, because Gomez was not a modest man. Alveolar surface area and gas exchange was one thing, but he was also interested in the aerodynamics, the way air flew from the trachea all the way out to the alveoli. And this occurs in an airway tree from the trachea through the bronchi all the way out to the acini where the alveoli are located. And this, of course, is different from alveoli because this is now a hierarchical structure, and it could not be approached by the same statistical stereological problems that we use to count alveoli. So we could use our counting method to estimate the number of terminal airways which had alveoli around them, and we found a number which then let us predict that there should be 23 generations of branching to reach that final stage. So that gave a framework for a model of the airway tree, which then I could complement with some measurements obtained on a cast, diameters and lengths and so on of airways. So we did all that. We had actually estimated the basics of the structure of the alveolar system, and we had the airway tree, and this resulted in a paper that Gomez and I could publish in Science under the title of Architecture of the Human Lung. But there was not yet much physiology in that.
Exactly. So you had all the basis from anatomy to do now finally we're supposed to do, namely physiology of the lung?
Yes, but the problem was we were missing the last crucial step. The point was that the methods I had available, light microscopy, did not allow me to resolve the structure in the capillary, the barrier, the plasma, the red cells, so that I could not analyze the morphometry of the final step of gas exchange that would be required for calculating the diffusing capacity that Gomez wanted to do on the basis of the Rautenfoster model.
So you had to go on now with electron microscopy?
Yes. In fact, in spring '61 was the time to, after 2 years approximately, to make a balance or check what we had achieved and what not. And I had to point out to Gomez and Cournand that I could not continue to the very end, that I would need electron microscopy for that. Cournand was immediately convinced of this need, and, but we must realize that electron microscopy was in its infancy in 1961. The basis for biological electron microscopy had led 9 years before by George Palade, and there has been some work, but not very much up to that time. So Cournand asked me, where could you do that? And I said, well, if I have a choice, uptown at Rockefeller Institute with George Palade. And that should in fact have been possible, because in the meantime I had become a career investigator of the Health Research Council of New York, a tenured lifetime position, which would allow me to work in any academic institution in the city of New York that would give me a place to work. So Cournand picks up the telephone, calls his friend René Dubos at Rockefeller, who arranges for me to meet Palade. And Palade tells me, well, you know, I do not take novices in electron microscopy. But since you are recommended by a Nobel laureate, I will make an exception.
So you switch your fields extremely and radically to do something completely new at that time that was brand new. Well,
I shifted my lab to Rockefeller Institute and found an entirely new environment. Bustling, it was the mecca of cell biology at the time. biochemists and electron microscopists studying cells on all sides. People coming by, they were also meddling a little bit in the new field of molecular biology. But Palade was a very generous mentor. He realized that the lung was actually at my heart, so he let me pursue my own project in his environment, studying the gas exchange apparatus in the lung at the electron microscope level, developing also the method for estimating the last step in this pulmonary gas exchange, namely the step from the alveolar air through the tissue barrier on the plasma into the red cells, as Gomez would need it for the last part of his model.
So, but if I recollect that correctly, your name became very famous in the Palade lab.
Yes, well, that was a serendipitous affair. I can tell you exactly the date. It was February 14, 1962. It was the 60th birthday of my father, who was in Switzerland. I was studying a rat lung, scanning through it, and since I was a novice in electron microscopy, looking at all sorts of structures, and I came upon a small pulmonary artery, which was a bit contracted. And when I studied the cells of the arterial wall, I saw in the endothelial cell a very strange structure, a stick 3 micrometers long, 0.2 micrometers in diameter, so very thin and long and straight. Something I had never seen, doesn't belong into a cell. But I had enough experience with stereology, knowing what it means to cut structures by a section, that I realized that to cut such a long structure lengthwise was an extremely rare event, would actually not happen. Most sections would be cross sections or oblique sections. So I looked around that section, and of course there they were, dozens of the cross sections of this little structure. Well, I showed it to Palade. He had never seen it, although he had looked at many vessels, and we went into his file of pictures, and sure enough, they were there. But they were very inconspicuous, 0.2 micrometer in diameter, not very spectacular. They had a membrane, they had some tubules inside. So
this little structure was the very famous Weibel-Palade body.
Well, that's what they became called in 1975 by Eric Jaffe, who identified them as the exclusive marker of endothelial cells in culture. But of course it was tempting at the time to abandon the work on the lung, working in this cell biology environment, and to concentrate on finding out what these damn little new things that we had discovered were, but it did not come to that.
Yes, this was the great break. So you left the US, having all these opportunities to come back to Switzerland. Tell me about this.
Well, the problem that by 1962 I had become an illegal immigrant to the United States, because my student's fellowship the visa for my student's fellowship had long expired. So I had to leave the United States and go back to Switzerland. Fortunately, Zurich had offered me an assistant professorship to set up the first electron microscopy unit at the university, which I accepted and did. But 3 years later, I moved to Bern because I had been appointed chair of the Department of Anatomy at the age of 35.
So that was very challenging. So you were first illegal, you come to Switzerland, and you get one professorship at the University of Zurich and the next one at the University of Bern, and you are 35 years old. So what did you do? That was a real big step in your career.
Yes, well, of course, the two jobs were very different. I mean, in Zurich, I simply set up my little research group. I had a new toy, the electron microscope, which I could use and help people. But in Bern, the situation was then very different. There I was to take on the responsibility for an entire institute, in fact, a bigger institute. Responsibility for teaching, for research, for administration. The institute in Bern was a quite beautiful building, but it lacked all installations and all the personnel to do any reasonable modern science. And not even for teaching, it was well equipped. So when I was nominated for this position, I looked at it and I wrote out a 60-page document, a plan to develop the institute over the next 5 years. The faculty approved that unanimously. But of course, the decision power was with the government, and what I had proposing was pretty heavy stuff. So after a marathon of different negotiations with all sorts of authorities of the canton of Bern, there was a final meeting in January 1966, and at this meeting there were 4 ministers, the education minister, finance minister, etc., and a few dozen administrators and the dean of the faculty. And I had to explain my plans. There was heated debate. And after about 2 hours, the Minister of Finance raises his hand and says, "Okay." And the meeting was adjourned. And 3 days later, the government had approved all my demands.
What was the reason that you just got an okay after so tough debates?
Well, a little later I got to know the finance minister personally, and I asked him, "Tell me, what happened on that ominous meeting that you suddenly said okay and everything was settled?" He said, "Well, it's easy. I liked you." Wow. That was politics.
Wow, these are politics, really. When exactly did you move to Bern?
Well, just 2 months after that ominous meeting. In the meantime, my wife and I had found a little house in a condominium, and Peter Burri, who had been my fellow in Zurich for about a year, came with me to Bern and was very important for setting up our new activity. I don't know how we actually managed to do all that, jumping immediately into teaching, trying to establish and refurbish the institute and setting up facilities for research, but somehow we did.
Excellent. So you had everything. You had the money, you had the institute, you have the people. Now let's switch gears and tell us what sort of projects did you follow for the next years to decades?
Well, of course, I wanted to make my institute a very modern institute, going with the current of time. So I had 3 topics that I had as major topics. The first one was to establish quantitative morphometry at all levels, all the way from the organism, and there we had the lung, down to the cell level, because I had, after all, the cell biology experience. And the second was to continue the studies on the structure of the lung and gas exchange function. And the third, of course, to try to see whether we could find out anything about these Weibel-Palade bodies.
Okay, let's start from sort of the beginning. So your first project, then on quantitative morphology?
Yes, well, I started first to develop the methods to apply stereology at the cell level, at high resolution electron microscopy levels. And this could be done particularly on the liver, which was the main object of cell biological studies at that time. And particularly, I could work with a group of basic scientists from the pharmaceutical industry who were interested in drug metabolism and the role the endoplasmic reticulum played in that. And this was a really fascinating line of work that kept us busy for the next 10 years, certainly. And in fact, I must say that the papers that have come out of this part of the work are my most cited papers, much more than those on the lung. And in 1970, a switch occurred in this project when Hans Howald, the director of the physiology lab at exercise physiology lab at the Swiss School of Sports, came and suggested that we could study the relationship between muscle mitochondria and maximal oxygen consumption in a group of top athletes, well-trained top athletes, compared with a group of medical students. And the work was to be done by Hans Hoppeler, one of our medical students, for his thesis. That turned out to be an important step in our further development because it brought us, of course, closer to the lung.
And Hans Hoppeler followed that basically for the rest of his life?
Yes, yes.
Let's switch now to the topic 2, gas exchange.
Well, the first problem was that we were missing one structure in the lung that had to exist, surfactant. John Clemens in 1962 had shown that the surfactant of the lung is a phospholipid film. And phospholipids should actually be fixed by our standard fixative osmium tetroxide. So why did we not see it? Maybe we simply washed it away. So we made a few experiments on rats. Perfusion fixed the lung through the vessels, and lo and behold, there it was. We had a beautiful two-layered system with a black film on top and swimming on a hypophase which was rather thin but separated the film from the epithelial cell. That was one part. We also studied different lung cells, but that's less important. But then I had a large series of projects on trying to find out the relationship between the lung structure and lung function by means of this model of the diffusing capacity, which we could calculate on the basis of the morphometric data compared to physiological measurements of oxygen consumption. So we did that, for example, on different sized lungs. Peter Geer studied dog lungs from the small Chihuahua up to the large Labrador, measured the diffusing capacity or measured the morphometry and then calculated diffusing capacity. And we went all the way down to the smallest mammal, the Etruscan shrew, which is about that size and weighs 2 grams but has the highest metabolic rate and an extremely dense lung. The other study was to see whether different energetic needs would have an effect on the lung. And there was one case, a medical student discovered once some Japanese waltzing mice in a pet shop. And I had her buy some of these beasts, and when we studied them, they really waltzed like mad. And the Swiss mice just looked dumbly around, and we measured their oxygen consumption and also then measured the diffusing capacity. And, well, oxygen consumption was twice as large in the waltzing mice, and so was the diffusing capacity. So it is matched.
So, but what about the human lung?
Well, the problem was that this was a very, very difficult problem. The problem was to obtain good, healthy, well-fixed, well-preserved lungs from humans. It took about 10 years until we had 8 reasonably fixed and well-preserved lungs that then could be studied in detail by light and electron microscopy by Peter Gehr and Marianne Bachhoven to obtain all the important measurements for calculating diffusing capacity. For example, they found that the alveolar surface area is nearly the size of a tennis court. So after, in 1978, we had all these data together and then could calculate the diffusing capacity, and there was a big surprise. The diffusing capacity we calculated from the morphometric data was 150 milliliters of oxygen that could be taken up per minute per millimeter mercury partial pressure gradient from alveolar air to the capillary blood. That was the unit. But the textbook value was 30. Now, well, the diffusing capacity of 30 corresponded to people lying on the bed in the basal state. But of course, the lung is hardly made to satisfy our oxygen needs while we are lying in bed. And indeed, when the measurements were made on exercising people, diffusing capacity values of about 100 were achieved, very close to our value, but not quite there. The question, of course, was how reliable our model and method for estimating diffusing capacity was. We didn't know at that time. It became clear only much later, in 1992, but I will just advance that because it's important. It was when Connie Shah from Dallas joined us. She had studied dogs running on a treadmill and had measured their diffusing capacity with carbon monoxide as a tracer, and found that as the dogs were running faster, blood flow increased, diffusing capacity would gradually increase. She called that recruitment of diffusing capacity. We could then study the morphometry of the diffusing capacity on the same dogs. And it turned out that our value was exactly the value that she had estimated by physiology at maximum oxygen consumption. So it seemed that the model was correct and the method worked. But we had a dilemma. From the comparative studies before, we had seen that in fact diffusing capacity was well-matched to oxygen consumption levels, but not when we looked at large and small animals in the allometric way. It seemed that the diffusing capacity was in fact proportional to body mass and not to oxygen consumption. So that large animals would have a much larger diffusing capacity than small ones. Well, that was a puzzle. I had no idea how to explain it.
Yeah, but we are going back now to 1975 with all these puzzles, with all these pieces of data. You just went away for a sabbatical?
Yes, I was entitled to a sabbatical and I decided I should let this puzzle rest for a while. Perhaps it would solve itself somehow. And I was still intrigued by the Weibel-Palade bodies and what they were doing. So I decided to take my sabbatical, go back to George Palade, who had in the meantime moved to Yale. Our plan was to isolate these rod-shaped granules from human umbilical endothelial cells who were particularly rich in these organelles, but we completely failed. These long rods simply broke apart by all fractionation procedures we tried, so we could not obtain a concentrated fraction to find out what was in these special granules. So we gave it up. Later, Denisa Wagner found out by an immunohistochemical study that they contained the von Willebrand factor, which is an important clotting protein, and it forms very long threads which in this granule are wound up in the form of these tubules. And it is an important protein for clotting, attracting thrombocytes to places of damage in the endothelial.
So how to overcome this frustration?
Well, it was indeed enormous frustration. I was not really used to have an experiment that I had well planned fail, but it does happen. So I spent a lot of time in the wonderful library of the Yale Medical School, looking around perhaps for a solution to my puzzle on the scaling of diffusing capacity. And indeed, I did come upon a paper by one C. Richard Taylor from Harvard University, who had looked at smaller and larger animals up to horses, measuring maximal oxygen consumption and finding their scaling. And when I looked at his graphs, they looked almost exactly what I had found for the morphometry of the diffusing capacity in our comparative studies. So maybe it was maximal oxygen consumption that was the solution instead of average oxygen consumption that we had measured as poorly capable physiologists.
So knowing Ewald Weibel view, the next step was obvious, immediately to contact Dick Taylor?
Yeah, I called him up from New Haven, and a few days later we met at the Harvard Faculty Club for lunch. And by the end of lunch, we had a complete joint research program ready. It meant to go to Africa to study wild species of various size, from about a pound up to half a ton. Well, we then also decided that perhaps, since we knew that VO2, or suspected VO2 max to be important, that we should also look at mitochondria in the muscle and not only at the lung. So that was a new project. It was actually typical for Dick Taylor. His grasp of a scientific problem was extraordinary. He could frame it into a project, and that was actually the heart of our long collaboration.
So basically you were already having a project, a new one to Africa, but your sabbatical was finishing.
Well, I first had to go home and see to my institute, and also applied to the Swiss National Science Foundation to have my two fairly large grants, which I had, one for lung and one for cells, to be joined into one for this African experiment, abandoning now the liver cells, but taking on the muscle mitochondria. And it was approved.
Wow, this is unusual.
Well,
so you went to Africa?
We went to Africa, yes. In 1977. I could only go for a few weeks because I had consumed my sabbatical. But Peter Gehr, who had done the large study on the dogs for allometry and was very well skilled with doing the morphometry, and he went there together with our chief technician to ensure high quality work. Dick Taylor went with a small team. And he had a huge treadmill shipped from Boston to Nairobi over a ship, and so we were all ready, in fact, to go. But then a drama happened.
In Kenya?
In Kenya. We were in Kenya, all ready to go, and then the drama came when Kenyatta's wife got a law passed through Congress to forbid all hunting of wild animals. Well, that looked like the end of our project. But then we had wonderful hosts there in Nairobi. One was the dean of the veterinary medical school, Jeffrey Malloy, a very capable comparative physiologist, a Maasai elder, a huge man who a few years later would get an honorary degree at Duke University and showed up in his full garb as a Maasai elder. And my team was in the veterinary anatomy department, and the chair was Wangari Maathai, a beautiful Kikuyu woman and very charismatic, and she would earn the Peace Nobel Prize in 2005 for her work on the Green Belt. So the problem was, within this environment, could we solve our problem? And Geoffrey Malloy now arranged for Dick Taylor to go and see Arap Moi. Arap Moi was the Minister of Justice and later became the President of Kenya. He explained the problem, and Arap Moi said, well, I cannot help you, but go and get your animals. If they put you to jail, I will get you out. Okay. We got our animals, and Moi did not have to get Dick out of jail.
What came out of that big African enterprise?
Well, in the 7 months that our teams were there, they studied 27 animals from about 1 pound up to 250 kilograms. They measured maximal oxygen consumption. Imagine that on a wild animal on a treadmill. Dick was very good at that. And measuring some additional physiological parameters. And then we obtained the muscle samples according to a certain scheme, and also, of course, had to fix the lung and sample the lung. And then this, of course, led to an enormous mass of material that then had to be shipped to Bern or to Harvard for further evaluation. And then the work actually began, which involved some 20 people, both in Bern and in Harvard, analyzing the physiological data and doing the morphometry and the electron microscopy on all these samples, with Peter Gehr responsible for the lungs and Hans Hoppeler for the muscles. The main problem in all this was really to maintain the communication between the groups in Bern and at Harvard. There was no internet at the time. We had telephones in the office of Dick Taylor and myself, so we could communicate in joint parallel group meetings, but that was about all. And then in 1980, we had a big final meeting here in Bern in this institute with both groups, those from Harvard and ours, to finally analyze all the data and put them together into a framework. We had established in the meantime a set of equations at the level of the lung, the circulation, and the muscle to relate the structures and the functions to maximal oxygen consumption as the common denominator for all these functions. And it was at this occasion that we invented the term symmorphosis, meaning something like balanced formation. We defined it as a principle of economic design of structures in terms of their functional performance. There should be enough, but not too much. And, well, we formulated it as a hypothesis that could be refuted or used to perhaps readjust our models if the conditions of the hypothesis were not fulfilled.
So with this African expedition, you solved the dilemma of scaling diffusion capacities?
Yes and no, or actually rather no and yes. No, because we confirmed the different scaling of diffusing capacity and oxygen consumption, even with maximal oxygen consumption. But it solved the problem because the symmorphosis concept told us that maybe we were missing a point in our analytical scheme. And the missing point turned out to be the alveolar partial pressure of oxygen the main driving force for getting oxygen from the air into the blood. It was assumed that this was a constant, but it could also be size dependent, and indeed it was, because in large animals, the terminal airways with the alveoli are much larger than in small animals, which means that as oxygen is moving in by flow and diffusion, it would end up with a lower mean alveolar PO2 in the large lungs than in the small lungs, which means that with a lower PO2, you need a larger diffusing capacity to get the amount of oxygen into the blood that is required. So that actually solved the problem, and it also showed the value of a hypothesis like symmorphosis that one did not necessarily have to reject if there were open points to be solved.
Yeah, that seems very obvious. Let's switch gears again, Ewald. Apart from all these scientific challenges and innovations, you have been also doing a lot of other stuff, like being rector of the University of Bern, and you have chaired a lot of other institutions. How was this possible?
Well, in retrospect, I also wonder how this was all feasible. But if you ask my wife, it was, of course, it meant a lot of sacrifice, both in private life and in leisure. But since I enjoyed all the things I did, I didn't suffer from it. So I just let it go.
Is that the secret?
Yes. And the point was, you know, I really never wanted to be a specialist. I always was interested much more than on the lung or on medicine. And so, in fact, it was a great time to be rector of the university, for example, because there I had to interact with all the faculties. I had to help solving problems in economics and also in the environmental sciences. I got also into touch with many people outside the university by having to represent it. So I met politicians, leaders in society. And then I realized that we had a big job of getting science out of the ivory tower and getting it into society. So as rector, I founded what is now called the Forum University and Society, a small group composed of half representatives of the university from all faculties and an equal number of people from the society, politicians or leaders in industry and so on. And that this has really been a success is that in the 30 years this has now existed, 2 of the political members of the Forum have been elected members of the Swiss government. Our current Minister of Justice is one example.
Okay. This is a nice success. And what have you done for the physiological field, societies and groups?
Well, I think the major job was to be president of the International Union of Physiological Sciences. As I had mentioned in the beginning, I was for 8 years on the council of the union, and then I was made president at the International Congress in St. Petersburg in Russia. And fortunately, this occurred after my retirement, because to clean up after that Congress was a really major job. Later, I became president of the Swiss Academy of Medical Sciences, where, of course, I had also a supporting function for physiology and all biomedical work, particularly with respect to ethics, as it occurs in all these disciplines. And, well, I suppose the greatest fun was to work on a number of foundations where I could help people active to do their work.
That's nice. So, and now thinking about the young medical student nowadays, your career has been basically driven by curiosity and motivation, also by opportunities and coincidence. Now, 60 years after finishing medical school, what would you recommend a young medical student if he wants to enter science?
Well, the problem is that things have changed. In my time, we were responsible for structuring our studies and curriculum. Now, of course, everything is determined by plans, and the student must make a special effort to retain the freedom of his thinking in this highly structured teaching system. So I would say the most important aspect would be to see that medicine is on the one hand a service, but on the other hand a science, and the science can contribute to improving the service. But of course, this depends now on finding weak spots in our system, and if as a student you find one, stick to it, try to discover it, and perhaps try to make a contribution to solve it. Even if it is a very small problem, it can turn out to be a big success in the end. Maintain your freedom of thought.
Ewald, thank you very much for your time, enthusiasm, motivation, and also mentorship. Thanks a lot.
Thank you very much, Max.